Hello and welcome to my presentation on initial responses to exercise. So, ATP is our main way of contraction. ATP stands for adenosine triphosphate. This is our main energy carrier. During exercise, ATP gets broken down to release energy for explosive action. This is broken down by the enzyme ATPase. This is great for short and explosive actions as ATP only lasts for 3 seconds. For example, from our Brighton Data University study, it is perfect for the standing vertical jump, as we are trying to jump as high as we can, and it only lasts for a short period of time. ATP also has a role in sliding filament theory. This is used to power the muscle contraction. ATP provides the energy for the myosin to pull the actin inwards. This creates the shortening of the muscle. As ATP has been broken down, it now needs to be resynthesized to be reused again for other sporting examples. So this action is done through the breaking down of PC. This is phosphocreatine, the lactic acid system, or the aerobic system. The system which resynthesizes ATP is dependent on the duration of the exercise, so it depends on how long the actual exercise lasts for. If the exercise lasts for around 3 to 10 seconds, the phosphocreatine system is used. Yet if the exercise continues and lasts from 10 seconds to 3 minutes, this is where the lactic acid system is used. Once the lactic acid system cannot be used anymore, as it's a waste product that makes our muscles sore, this must be replaced and ATP is resynthesized aerobically. We collected data from the Brighton University, and this data shows that the vertical jump lasts no longer on average than 0.58 seconds. And the longest jumps in terms of time in the air was only 0.71 seconds. The vertical jump requires a short, intense burst of energy for a quick contraction. This is a quick and short contraction, which ATP is the dominant system to use for this. The CNS stands for the central nervous system. This system consists of the brain and the spinal cord, which are responsible for passing impulses quickly around the body. The other system is the PNS, which stands for the peripheral nervous system. This is made of neurons in the body. The PNS takes impulses to and from receptors and effectors. The information from the receptors travel along a sensory neuron as an electrical impulse and is sent to the central nervous system. At a synapse, the impulse travels quickly through the CNS and over another synapse onto the required motor neuron. This impulse then travels along the motor neuron to the axon terminals in an effector muscle. This is vital with muscle contraction, as this system helps us to respond to the environment and make the right choices. Neurons are made of an axon, which is a long thread with the impulses conducted on. The myelating sheath is a fatty substance which surrounds the axon. This speeds up the speed of the impulse, and the saltatory conduction is the involvement of the impulse jumping from one node of Ravier to the next. You can see this on the graph here. The nodes of Ravier are the little gaps between the myelin sheath. The node of Ravier is the interval between the lumps of myelin sheath, like I just said, and the saltatory conduction speeds up this process. Twitches are a problem regarding recruitment of motor unit. A twitch can occur when the muscle is stimulated before it has the chance to fully relax and rest. Many twitches which have been built up can lead to a tetanus contraction. We can see this here. This is the tetanus contraction. A tetanus contraction is a huge contraction without any relaxation. By looking at the graph, we are able to see how the twitch contraction happens. If there are two stimuli close together, there will be a twitch contraction. This is the two stimuli here, and that's the twitch contraction. And if there are many stimuli, this leads to a huge contraction, as tetanus, which I've already shown. In our body, we have a fibre called the fast glycolytic fibre. This is the main bulk of our body's muscle mass. These fibres produce the most powerful contractions within our body. They have a few mitochondria and blood capillaries, which means they don't work aerobically very well. The fibres contract very strongly and quickly. This means they are also very useful for the vertical jump, which explains, like, if they're used in if they're not used aerobically, they're not going to be used in long distance events. A motor unit made up of a motor neuron and skeletal muscle fibers, which are innervated by that neuron. The group of motor units work together to produce the contraction of a muscle. The higher number of motor units recruited, the stronger the contraction will be. The size of the motor unit also depends on the contraction. If the contraction needs to be stronger, the recruitment of motor units will be larger, meaning the motor unit overall will be much larger. The size of the motor unit changes during exercise. It gets much larger as we are working our muscles, and this means there will be a higher contraction because of this. Because of this, more motor units are recruited, increasing the size. So the neuromuscular junction is the site which electrical impulses jump from the motor nerve axons to receptors on the muscle fibers. This process begins with an electrical impulse traveling down the axon of the motor unit. This impulse causes the calcium channels to open, which triggers the vessels containing neurotransmitters to cross the synaptic cleft. The synaptic cleft is the gap between the axon and the receptors. This is the gap. The neurotransmitters cross the synaptic cleft with acetylcholine. This chemical eases the transmission of the neurotransmitters from the axon to the receptors on the fiber. This then fires the impulse to the muscles. This is an image of the depolarization. Depolarization occurs in the four chambers of the heart. The sinoatrial node on the wall of the right atrium initiates depolarization in the right and left atria, causing contraction. This wave of depolarization then spreads down to the ventricles. So, the muscle spindle apparatus are receptors within a muscle fiber which detects change in the length of the muscle and how much the muscle is being strained from the stretch. They send information to the central nervous system through afferent nerve fibers. This information gets processed and the brain determines the position of body parts. The intrafusal fibers detect stretch. This causes the stretch reflex sometimes when necessary. This reflex is where the muscle contracts to the response of stretching within the muscle. This process is autonomic. The other fiber is the extrafusal fiber, which is this bit here. These fibers are the general muscle fibers which are the bulk of the muscle. The stretch-shorten cycle involves the stretched muscle to immediately shortening after it has been stretched. An example of this would be the vertical jump. Our quadriceps are resting first, but as we jump down they contract, causing the stretch. As we jump back up, this is immediately becomes shorter and returns back to normal length. Elastic energy is the energy created when objects are stretched or compressed. The stretch reflex produces elastic energy. This is how the stretch-shorten cycle occurs and is able to take place. Plyometrics is taking place to improve these reflexes. This type of exercise makes the muscles use their maximum force in short intervals and the idea of improving power. Plyometrics can be used in the vertical jump as there's a short burst of powerful contractions to reach a maximum height. By looking at the data, we are able to see that the counter-movement jump is much better for gaining higher height in vertical jump. This is due to the stretch-shorten cycle combined with elastic energy. This causes strain on the muscles which are exerted through the height of the jump. The Golgi tendon organ is a sensory receptor organ which detects change in the muscle's tension. This is placed between the origins and insertion of skeletal muscle fibers into the tendons of skeletal muscles. Although during the contraction the muscle's tension is increased, the alpha motor neurons in the spinal cord inhibit the muscle. This impulse to inhibit the muscle is from the central nervous system. This is sent to the spinal cord and then directed to the muscle through an afferent nerve. The Golgi tendon organ inhibits this due to too much strain on the tendon during the contraction. Autogenic inhibition is one of the Golgi tendon organ's function. This is where there is a reduction in excitability of a contracting muscle. This is caused by the Golgi tendon organ inhibiting the muscle. The lactic acid system is known as anaerobic glycolysis. This is where glucose is broken down by enzymes and turned to lactic acid where there is no oxygen. Glycogen stored in the muscles is broken down by the enzyme glycogen phosphorylase and turned into glucose. Through this process, 2-ATP are used. This glucose then gets broken down even more into pyruvic acid through the enzyme phosphofructokinase. If no oxygen, from this point onwards, it is turned into lactic acid through the enzyme lactate dehydrogenase. Lactic acid is the product of anaerobic glycolysis. This system can be used through short, intense exercise. The system only provides us with enough energy to last us between 10 seconds to 3 minutes. Yet, as lactic acid is a waste product, as it makes our muscles sore, it must be removed from our system. The impact of using this system is that our muscles become fatigued due to it. Then moving on to the phosphocreatine system, which is this part here. The PC system stands for phosphocreatine. This is found in the muscles of any organisms. The role of the phosphocreatine is to resynthesize ATP to replenish the system. We do it naturally at rest. We do not need oxygen to restore this system as a chemical fuel source. Phosphocreatine is broken down by the enzyme creatine kinase. This is a high energy volume which is broken down. This energy is used to resynthesize ATP. This is where ATP gets turned into ADP plus P, and it binds the ADP plus P back together to make ATP. This system is useful because it lasts longer than the ATP. We are able to use this system for 10 seconds of muscle contraction. It allows for a high intense work. This is perfect for high intense events such as 100-meter sprint, yet the disadvantage of this system is that it only lasts 10 seconds, and for some events this is not long enough to constantly use this system. By looking at this graph, we are able to see the OBLA. This is the onset of blood lactate accumulation. This is measured at 4 millimoles. We can see this here. This is measured right here. On this graph, this is around 3.887 millimoles, mmol. The power when the OBLA sets in is around 190 watts. On this set of data, we are also able to see the lactate threshold of the person. The lactate threshold is where the rapid increase in lactic acid. On this individual set of data, this is 1.212 of lactate with a power of around 137 watts of power. This is here. This is before the lactate suddenly increases and has a massive increase. We collected this data from the set of data we got from the Brighton University study. The athlete was on the bike for a set amount of time. As the athlete went on, the power increased. We are able to see where the power increased. When the power increased, the lactate began to increase, and so did the heart rate of the athlete. The Valsalva maneuver. We use this when we are lifting heavy weight. It's essentially trapping the oxygen we have breathed in the stomach. This creates lumbar spine support, intra-abdominal pressure, which also helps with the sport, and we use this regularly with our high demand weightlifting. This process would have been used during the tests which were taking place at the Brighton University. These tests would include 1 rep maximum test of the leg press, chest press, and the back pull. All these exercises include using a heavy weight and using our maximum effort to complete these exercises. For respiratory dynamics, we used the VO2 max test, which brought us readings of their absolute oxygen uptake during exercise. This test was where the athlete was breathing into a Douglas bag for 30 seconds at the end of the 3-minute stage. We can calculate our ventilation by multiplying our breathing rate by our tidal volume. For the breathing rate, and the normal values are between 12 to 15 breaths per minute. This is at rest, and for a tidal volume, this is around 500 milliliters. The submaximal/moderate exercises can increase to 30 breaths and 1,500 milliliters of oxygen. For ventilation, the resting values are 6 liters per minute and rise to 45 liters per minute as submaximal exercise. To estimate our athlete's breathing rate and our tidal volume, we can look at our athlete's ventilation. This was around 39 liters per minute when they were at the peak of their exercise. From this we can calculate the others. This means the breathing rate must be around 28 breaths per minute, the tidal volume would be around 1,400.